Thin layer chromatography. Each of the dyes in the mixture has a different affinity for the silica on the plate compared with its affinity for the solvent drawn over the plate by capillary action. We can stop the separation by removing the plate from the solvent. In thin layer chromatography, we may be interested in the number of components in the mixture or in showing that a particular substance is present. But suppose you want to isolate the components. You would need to work on a much larger scale and be able to collect each separated component. Both are possible with column chromatography. Again, you can use silica as the stationary phase, but the solvent, the mobile phase, is flowing under gravity. If only separations in organic chemistry were as simple as this. In the first place, the components are not usually colored. In TLC, you can find out what's happened by using a color-forming reagent. In column chromatography, you can use a similar method to test each of the separated fractions. Secondly, you may want to know how much there is of each component. That's difficult with TLC, and with a column you would probably have to evaporate and weigh each fraction. Thirdly, these methods are limited to simple mixtures with, say, up to 5 components. But many samples, biological, forensic, medical, food, and so on, are much more complex. What we want is a stationary phase with much better performance. If we reduce the size of the particles, there is less band spreading, and we get much cleaner separations. But now gravity is no longer adequate to pull the mobile phase through a column. What's needed? Pressure will do the trick. But how can you get a sample into a closed system under pressure? We use an injection valve, where the sample is first loaded in a tiny loop of tubing and then transferred by a switch into the high-pressure system. After separation, we want to not only detect the components, but also find out how much of each is present. We need some property of the components that distinguishes them from the mobile phase and that can be expressed as an electrical signal whose strength is proportional to concentration. Well, the commonest type of detector uses the absorption of light. The electrical output from the detector is monitored against time, either on a chart recorder or as data to be stored in a computer for manipulation and display at a later time. Well, this type of liquid chromatography is far removed from where we began, but it has solved our 3 problems: detection of the sample, quantitative measurement, and above all, it is high-performance liquid chromatography, or HPLC, and it's capable of some very impressive separations. Well, now let's look at the instrument itself. Here's the solvent reservoir, the mobile phase. This is drawn into the system and forced by a pump to flow under pressure at a pre-selected rate. The sample is introduced at this point using a valve injector and arrives quickly at the top of a column filled with the stationary phase, for example, microparticulate silica. After leaving the column, the separated components pass through the detector, in this case, a single wavelength detector. You can dial up the wavelength you want to use. But how would you choose a wavelength? Well, clearly, it must be one that's absorbed by the components, but not, as here, by the mobile phase. The mobile phase must have an absorption cutoff below the wavelength for detecting the components. This presents no problem with, say, hexane, which is transparent virtually to the limit of the instrument. But if, for solubility reasons, we have to use, say, dichloromethane, the cutoff rises to 235 nanometers. For toluene, it would be as high as 285 nanometers. So, using a wavelength absorbed to some extent by each component of the mixture, but not by the solvent, the detector responds with an electrical signal proportional in strength to the concentration of the compound passing through. And against elapsed time, this gives a peak on the recorder. The separated components therefore appear as a series of peaks, or a chromatogram, and the complete instrument is called a high-performance liquid chromatograph. Let's now use this chromatograph to analyze the mixture of dyes. First, we must select the parameters for the separation. In practice, this is based on experience and trial runs. The dyes are fairly non-polar compounds, so we are using a column packed with 5 micrometer silica. And our trial suggested dichloromethane for the mobile phase. The flow rate will be 1 ml per minute. The dyes absorb well clear of the 235 nanometer cutoff for dichloromethane, and we'll select 340 for the detector wavelength. Now we just set the recorder integrator at a convenient speed and sensitivity, and we're ready to inject the sample. A suitable sample concentration was determined beforehand, and we are using a 10 microliter loop in the injector. The loop is flushed with sample with a syringe. And then, on rotating the valve, we start the run. Here's the first dye, and after 10 minutes, the chromatogram is complete. Let's compare it with the TLC we saw earlier, which ran from right to left. Which peak do you think corresponds to the slowest moving spot, the blue one on the right? Well, the slowest moving dye on TLC is the one most highly retained by the stationary phase, and this corresponds to the last to emerge from the HPLC column. So, using the same stationary phase, the sequence is reversed in relation to the starting point. As with TLC, you could use HPLC to check the possibility of a known substance being present. In fact, the best way to do this is to add the known substance to the mixture and see if one peak becomes bigger while perfectly retaining its shape. The chromatogram has more than graphical information. The peaks are listed by retention time, and then the area of each peak is given together with its corresponding fraction of the total area of the chromatogram. As peak area is proportional to concentration, you can use HPLC to find out how much of one or more of the components are present. Suppose, for example, you wanted to find out the molar concentration of this substance in the mixture. If I gave you a pure sample and the solution of dye mixture, how would you use HPLC to determine the concentration of this in this? You may like to stop the tape and think about it. You'll find an answer in the video notes. The separations you've seen so far rely on differing affinities for hydrated silica relative to the less polar mobile phase flowing over it. Good separation depends on the nature and extent of polar groups within the molecules of the mixture. Here we're indicating polar by blue and non-polar by yellow. The more polar the molecule, the more it is retained by the polar silica, and the slower is its elution by the comparatively nonpolar mobile phase. Now, work in this so-called normal phase situation is limited by sample solubility. If we could use only a nonpolar mobile phase like chloroform over a polar stationary phase like silica, most water-soluble mixtures like biological or medical samples couldn't be analyzed. We would like to use water as our mobile phase, but then the stationary phase would have to be nonpolar. This means reversing the phases. Nonpolar stationary phases are now readily available. Most are based on silica, but the oxygen atoms on the surface are chemically bonded to long hydrocarbon chains. The few OH groups that escape this process are then capped with the trimethyl-silyl group. Now, when we have a mixture of polar organic compounds dissolved in water, it is those with the most nonpolar parts that are retained. In most other respects, the chromatographic principles and instrumentation is the same as before. But think of the advantages. We can look at biochemicals like amino acids, peptides, carbohydrates, and using water-miscible organics like acetonitrile and ethanol, we can analyze a very wide range of polar organic compounds. These mobile phases are less volatile, less toxic, and less corrosive than normal phase solvents, and their lack of absorption above 200 nanometers is very convenient. Also, many separations can be improved by adding water-soluble modifiers like buffer salts and ion pairing agents. In fact, by far the majority of HPLC separations nowadays are carried out in reverse phase systems. Here, for example, is a mixture of small peptides dissolved in water which we couldn't look at by normal phase. The column is silica bonded to chains of 18 carbon atoms, and we are using as our mobile phase 5% acetonitrile in water containing a small amount of heptafluorobutyric acid to help the separation. The flow rate is 1 ml per minute, and as these peptides are weak absorbers, it's handy to be able to use a low wavelength like 215 nanometers. This catches the tail of their absorption in the far UV. Sample into the loop, and in goes a few micrograms of mixture onto the column. And 20 minutes later, the peptides are nicely separated, and if wanted, we could find out exactly how much of each one is present. Analytical information like this from a microgram of sample is now routine. But suppose you wanted to isolate one of these peptides. Clearly you couldn't handle tiny amounts like this. You would have to scale it up, at least to milligrams, and you may want to separate grams of material. How can HPLC cope with that? Well, first we have to consider the sample size. Here is the analytical separation of peptides. How far can we increase the load on the same column and still maintain a useful separation? If we load it not with 20 micrograms, but with 1 milligram, the separation is still acceptable. Now, using a similar stationary phase, but a much larger column, you can load up with 25 milligrams per injection and still expect a usable chromatogram. In addition to the larger column, you would need a larger loop injector and larger scale plumbing. Well, this would allow you to separate 25 milligrams in a single injection, and to separate half a gram of mixture, you would need to inject 20 times. For much larger samples, you could use even larger columns and automate injections from a large reservoir sample. At the other end, the key component is the fraction collector. This directs output from the column into containers at times set to coincide with the peaks we want in the chromatogram. Then, to recover each separated component, we simply evaporate the mobile phase. In the next part of the tape, we're going to look at your experiment on cycloisoleucyl isoleucine and how HPLC can be put to work. You should carry out the experiment first, but before that, please use the video notes to review the tape so far. A solution of isoleucine in refluxing ethane 1,2-diol. Remember, we expect this to provide the cyclic dipeptide cycloisoleucyl isoleucine. You've filtered the crude product and after recrystallization from ethanol, obtained a white solid. Let's run its infrared spectrum. The best way is to make a disc of the sample mixed with potassium bromide, because this salt is transparent in the spectroscopic region we're interested in. We need less than 1 milligram of sample in 100 milligrams of potassium bromide. These are well mixed and carefully ground to a very fine powder. The mixture is loaded into the body of a stainless steel die and tamped down with the plunger. Assembly of the die is completed by inserting first a machined steel pellet and then the plunger. The whole apparatus is taken to a press where the sample is subjected to high pressure for a few minutes. Dismantling the die releases a disc which should be no more than slightly opaque. The disc is mounted in a special holder and then supported in the sample beam of an infrared spectrometer. And here's the spectrum of the product. And that looks pretty good to me for the compound we want, but you can check on that later. Remember, we expected this to be one of 3 stereoisomers of cycloisoleucyl isoleucine, and we planned to treat it with base in the hope of getting a mixture of all 3. Here we're using sodium ethoxide in ethanol. For workup, we first add an amount of hydrochloric acid calculated to neutralize the base, then separate the cyclic dipeptides from the sodium chloride produced by extracting them with chloroform. We'll analyze the chloroform layer by TLC. We want to compare it with our product before isomerization and with what we know to be just one of the stereoisomers, the SS isomer, because we synthesized it stereoselectively from S-isoleucine. We visualize the plate by spraying first with hypochlorite solution and after 20 minutes with a mixture of starch and potassium iodide. Well, what do you make of this? It appears we had the 3 isomers there all along, and that treatment with base was not necessary. Or was it? Remember, the idea of the project is to isolate each of the isomers, so we need to know not only whether all 3 are present, but also in what amounts. For that, we're going to use HPLC. Every new HPLC separation requires development work. After several trials, we found the best conditions for the stereoisomers were these. First, a reverse phase column C18 on 15 micrometer silica. Second, a mobile phase of 20% acetonitrile in water. No modifiers are needed. Third, our instrument parameters should be flow rate 1 ml per minute, detector wavelength 215 nanometers, which is very low, but the compounds absorb only at short wavelength. Let's run first the mixture obtained after treatment with sodium ethoxide. The sample is a saturated solution in ethanol, and we're using a 10 microliter loop. These three peaks here are probably our three isomers, and this less well-retained material, decomposition products. When we ran the SS isomer, we found it corresponds to the last of the three peaks in the mixture. When we ran the product obtained directly from isoleucine, that is before adding base, we saw again that all three isomers are already present, but now we can see that they are present in useful amounts. You can examine this data more closely in the notes later. But now we must check that these are the three isomers by isolating them using preparative HPLC. We want to separate 1 gram of the mixture obtained by heating isoleucine. We use this C18 reverse phase column for the analytical work. Here's a suitable preparative column of similar material. But in trying to scale up the analytical experiment, we ran into a problem. The solubility of the mixture in water-miscible solvents is so low that we would need 20 litres of sample to separate 1 gram of mixture. And this would mean thousands of 2 ml injections and an enormous volume of mobile phase, not to mention weeks of continuous use of the chromatograph. So for the preparative work, we decided not to use reverse phase chromatography, but normal phase. Here's the analytical chromatogram obtained using a column of 5 micron silica and dichloromethane as the mobile phase. Using dichloromethane meant increasing the detector wavelength to 238 nanometers, where the cyclic dipeptides absorb very weakly indeed. Fortunately, the detector has adequate sensitivity for the job. Now our 1 gram sample will dissolve in 300 ml of dichloromethane. So for complete separation, we would need only 150 2 ml injections. Let's put one of these injections through with this preparative column of 15 micron silica and dichloromethane. Our flow rate is now 25 ml per minute, and the detector is set at 238 nanometers. In goes 2 ml of solution, and 10 minutes later, the chromatogram is complete. The high loading has certainly affected the shapes of the peaks, but the resolution isn't too bad. We'll take our cuts for collection here, here, and here, and the other parts will go to waste. This means setting the collection program at the corresponding periods of time and carrying out 150 separations with an automatic sampler. Well, we've stopped this after 40 injections to see how it's going, and our 3 isomers should be in these 3 sets of flasks, the rest having been sent to the waste bottle. Let's check purity with analytical HPLC. Here's the peak 1 sample, and that's not bad at all. Now the peak 2 and the peak 3 sample. So our 3 components are being nicely separated from the mixture, which look like this. Well, this is acceptable, so we let the separation continue, and from 1 gram of the mixture we were able to recover 400 milligrams of the substance responsible for the first peak, 120 milligrams of peak 2, and 100 milligrams of peak 3, which I can tell you is the SS isomer of cycloisoleucyl isoleucine. We have provided you with spectroscopic data for the separated samples, and we hope you will be able not only to prove that these substances are the other 2 isomers, but also which of them is the SR isomer of cycloisoleucyl isoleucine and which is the RR isomer.